All right, so I'm Rachel Washburn. I run the Center for Geohealth and Applied Omics Studies, or the CHAOS Lab, at the Kentucky Geological Survey. We're based out of University of Kentucky. So my background is actually in biomedicine. I have a PhD in biomedical sciences, which how do you get to mapping in geology? It's like a nice wavy route. I guess I could have Emily help me map that out, and that could be a presentation one day. But basically, I was always interested in interdisciplinary work. So the intersection of where medicine meets geology, meets geospatial integration, meets chemistry, and that's kind of what this map is. So Microbial Cartography in Kentucky, this is our new product. We made it this year, and I'm going to tell you about the good, the bad, and the bioproductive of it. This talk's kind of an overarching explanation of what it is. It's not too technical. If you do have technical questions, I'd be happy to put you in contact with our geospatial expert. But as far as applications go, that is my specialty. So why map microbes? Well, there's a lot of maps of biodiversity out there, but what's interesting is there's not really any geospatial maps of microbes. And microbes make up over 99% of all biodiversity. The only microbe maps I've really seen have been in disease models or like in the human body, and there's a ton of those. And we call those microbiome maps, and that's where we're getting the Kentucky Microbiome Map from. So we have this huge amount of biodiversity that hasn't actually been mapped out. And again, there's not maps of where we can find these microorganisms in nature. Like we'll see it in clinic, in bodies, in animals, but not quite in nature. So as a biomedical scientist, and I specialized in microbiology, that was a good fit for the Kentucky Geological Survey to kind of see how geologic processes are affecting these microbes. And the first step is to know where they are. But this map is more than just, gee whiz, that's really cool, nobody's doing it. There's a lot of different applications for it. A big one is public health and water quality, so things like tracking E. coli as they move through fluvial systems, our rivers and streams, knowing what's in our soils, and being able to monitor things for harmful algal blooms and recreational waters. Agriculture is very interested in what microbes exist naturally as far as a good microbiome to support robust crops or in carbon cycling and nutrient balance. As far as geology goes, microbes actually play a big role in cave formation and in weathering. So we are seeing microbes as they're influencing how landscapes form. Human health and medicine, this is really where microbiology shines. Usually if you think microbiology, you think diseases and illnesses. And by having a geospatial map of the microorganisms in the environment, it can help us identify any potential emerging pathogens so we can be prepared for future infections and also mitigate those things. And my favorite application, there we go, is looking at microbes as a resource. A lot of times we think microbiology, microbes are bad, they get us sick, but there's a lot of stuff they do to keep us healthy and that we can use them for. So things like bioprospecting, looking at microbes for any kind of enzymes or chemicals they make that we can use for new medicines, for industrial practices, so natural product discovery. We can use them in biomining. We can use the chemicals they're making to try and pull critical elements out of deposits. So this is kind of the future of geospatial-based microbiology. All right, there we go. Okay. So this map, we wanted to create this open access platform that can be used by anybody, academics, clinicians, industry, government, and even just the standard person who thinks, hey, maps are cool, and I think microbes are neat. We want this to be available, open access, free of charge. The hope is that this will help us foster collaborations, both by providing people preliminary data for grants, preliminary data to have new studies and collaborations, startup ideas, with just this ready-to-use data set. As of the time before this map, there wasn't a single platform that existed that integrated a bunch of different disciplines. So this map is integrating microbial data sets, geological data sets, soil hydrology, and chemical data sets into kind of a one-stop shop. And it is the very first statewide integration of these different fields where we're putting the information out there and geospatially mapping them. So the big impact for KGS is we are putting Kentucky at the forefront of geospatial-based applied microbiology. All right, so how did we make it? What is actually contained in this map? And I'll show you the map after I tell you how I made it. We have our primary data sets. This is stuff coming from the Kentucky Geological Survey. So this is stuff, I'm primarily in charge of it. This is stuff I'm going into the field. I'm going into caves or into like fluvial areas, into soil areas, and collecting things like sediment, soil, water, and I'm swabbing things. In addition, we're also collecting things to see the chemical composition of the area. We're collecting atmospheric data, and we're looking at the physical conditions like, is it wet? Is it dry? Is it humid? Is it not? Is there light? Is it dark? These are all the different kinds of things that we are collecting when we come out there. So that's kind of our raw data, and then we're processing it, so we're trying to make it more usable. Yes, you can access that raw data, but you'll also get more processed data, like what DNA did we actually find in what samples? Metabolites, that's a fancy word for chemical arsenal. It's just what the microbes make that we want to use. So we're looking at that too so we can identify it. We are also looking at environmental metadata and putting all that available. And we're further analyzing molecularly to see not just what DNA or transcripts or genomes that we see there, but what's the taxonomy, which is just what's the family of microbes? What are the specific species we're finding? Those metabolites, how do those compare to chemicals we already know? And then on the other side with geochemistry, we're doing a solid geological analysis. So what's the mineralogy? What elements do we see? What anions, cations? And we're looking at all that together, and then we are mapping that geospatially. Our secondary data sets are going to come from collaborators, and they're going to come from literature. So we have collaborators at Eastern Tennessee University and Appalachia State that have been giving us microbe sets that we are putting on the map right now. And then literally this map started while I was waiting to get access to a number of areas in Kentucky with permitting, and I just did a literature search. So we have people that are constantly looking over the literature to keep this map updated. And the search criteria is it has to be a microbial study and it has to be in a natural environment, so no clinics. That's really the only thing that we're not looking at. And we take all that and we validate it and verify, make sure it fits within the scope of the map, make sure it's peer-reviewed, and then we also geospatially reference that. So you get all of this information, and it is our open access Kentucky Microbiome Map. So that's the recipe. So this is what it looks like. And if you'd like to follow along, I have the QR code for our map, and I will walk you through kind of how this map looks. So when you open the page, you're brought to an abstract that describes what the map is for and gives you a citation. Then you get this heat map with a number of pins, and you have the legend up there, so it describes what the heat map is. You can turn on and off layers right now. And each of these blue points, those are going to be environmental testing sites for different rivers and streams. And it will tell you what the coli was, what the pH was, whatever was tested at that time. If you click on the heat map, it'll tell you the E. coli and how it compares as it's been tested over time. And you can turn on and off layers. So these pins here are the actual study pins. Each one of these pins represents a study that was done in Kentucky. It gives you the whole taxonomy of the microbes and all the microbes that were identified, so you could see what kind it was, what kind of sample it came from. Was it karst? Was it soil? Was it a swab? Was it water? And then you get the whole reference and a link to the paper itself. So instead of having to do a whole bunch of Google Scholar or PubMed searches, it's all already there. Your reference, your citation, and sources. And you can use this to track things like white nose syndrome. You can look at where other coliform bacteria, if you're interested in environmental sampling, and we can use it to pinpoint sites for bioprospecting. And right now, that shows you 3 layers of the map. We're currently working on expanding the searchability of the map itself. So this is still relatively new. We are considering it a living map, and there's more data that will be coming. So I talked a bit about microbes and metabolites, but we're also going to add in any genetic information we find in cellular structures, so that microbiological information. And geologically, we're adding this in, so you'll know the bulk minerals, you'll know any heavy metals, any toxins that were there, and the geological history and the formation it was found in. And then all the atmospheric data is going to be added as well. So temperature, humidity, light, anything that was taken. Gases, we measure methane and radon a lot at the Geological Survey, so if we measure that in conjunction with sampling, you'll get that too, as well as the date and time of when those samples were collected, so you can look at it temporally, spatially, and everything in between. And the goal is that this is a living atlas. So we're layering all this information that doesn't usually get layered. Metagenomics, metabolomics, geochemistry, microbiology, all together in one searchable map. And we're making all the layers searchable. You can filter through them, so you'll be able to look, if you're interested in specific microbe species, you'll be able to look at, I'm only interested in staph, that will be a layer you can search. I'm only interested in cave environments, that will be something you can search. So we're making it very user-driven, and if you do use this map and you have any suggestions on what you want to see, let us know, and we will find a way to make it better. We're hoping that this helps us expand collaborations across multiple disciplines, especially in geohealth. There's a lot of silos in science, but geohealth is a mix of so many disciplines, and we need to collaborate with these disciplines. So hopefully that's what this map is going to help us do. And though it is in Kentucky, we are taking data from other areas too and applying it to the map, but Kentucky is unique in that it's like a convergence of a bunch of geological formations together, and actually the environments within Kentucky make really good models to study areas that are similar worldwide. So Kentucky's a good model study area. The whole goal is to make this a geospatial discovery engine for natural products, for informing public health, and for fueling future research. So, again, towards that living atlas, microbes are as much a part of the Kentucky landscape and of the landscape in general as our rocks and rivers, but they're invisible, so we don't think about them. And that's what we're trying to do. We made this map to be an atlas of the invisible. The map is live now. It's not perfect right now. We're still making it more searchable, more filterable, but you can use it now. You can reference it now. It already has links to citations. You can use it for sure. And the end goal is for it to support a wide range of research applications. So this is just a short list, but really, I feel like there's a lot of uses. We could talk about uses all day. But yeah, we just want to try and see if we can expand our collaborative network and get a more systems understanding of what's happening in the world. So I want to acknowledge our collaborators, especially Doug Curl, who did the GIS side of this. I put together all the microbial data sets, and he's helping me map and create the map itself. Emily's helping us make a static map of the E. coli levels. And I need to acknowledge our community partners. They're the reason we can sample where we are. We're kind of unique at the Geological Survey in that as a survey, we can go sample a lot of places without really having to explain why we're doing those places, because we got a survey. We're a geological survey. So that's why we can create something like this. And then our funding with the NIH and the Office of the Vice President of Research. And I definitely have to shout out to our Kentucky Watershed Watch. They're a group of citizen scientists who have done all of that E. coli testing. So use our map. It's live. Contact us if you have any ideas or if you want to collaborate. We're active on social media, and we post updates as to what we're changing with the map and how we've updated over time. Thank you. ChatGPT. I said, make a microbe image like the good, the bad, and the ugly with a standoff, and that's what it did. It was perfect. Oh, it took like 5 different prompt revisions, but I feel like I'm getting better at it. We absolutely could put that onto a map. And in fact, if they're interested, if they talk to us, we can talk about how to make that happen. A number of our collaborators want to wait till they get their first paper publication out where they have to publish the data set. But yeah, absolutely, we could put that on there.